hello to all the AP, EAPCET aspirants. Hope you are doing great with your preparation, beta. In this session, we are going to revise all the concepts of genetics in just 30 minutes. So stay tuned. In the chapter Principles of Inheritance and Variation, we have to focus on the model organisms like pea plant and Drosophila melanogaster, which is taken by Morgan. And we have to look into the monohybrid cross, dihybrid cross, Mendelian principles, exceptions of Mendelian principles. Coming to the numerical part, we have to be aware of the all ratios and the formula to know the types of gametes, zygotes, number of phenotypes and number of genotypes. And next we have to be aware of chromosomal theory of inheritance, chromosomal mapping and sex determination, lastly Mendelian disorders and pedigree analysis. Let us remember and recall all of them in just 30 minutes. Stay tuned. So yes, when it comes to the model organism, that is Pisum sativum, on which Mendel has worked for around 7 years. Mendel has worked on it for 7 years on the plant Pisum sativum. This has taken as model organism because it is an annual plant and it can show clear contrasting characters and it can easily go through self-pollination, cross-pollination can be easily conducted, and this shows stable trait of inheritance. And then if you see, Mendel has taken 7 traits or characters, 7 traits or characters he has considered from Pisum sativum, of which how many varieties is your question? Then you have to opt for 14 varieties. Okay, 7 traits or characters and 14 varieties you have to opt for, beta. What are those 7 traits or characters? First is seed shape, round is dominant, wrinkled is recessive. Flower color, purple is dominant color, white is recessive. Flower position, axial position is dominant, terminal is recessive. When it comes to seed color, it is yellow color dominant, green color recessive. Pod color is green dominant, yellow recessive. Pod shape, inflattened is dominant, constricted is recessive. Most important, which we are considering for monohybrid cross, that is plant height, tall is dominant, short is recessive. So what we have to remember, all the contrasting traits, which is dominant, which is recessive. Everything should be in our mind. Numericals can be asked taking any pair into consideration, beta. So keep all the varieties with the dominant and recessive traits in your mind. Okay. So yes, now when we go to monohybrid cross, where single hybrid is being considered, only one trait is there, that is tall. Okay, that you can say plant height. Plant height is the only trait which is considered for monohybrid cross. Whereas tall is dominant, dwarf is recessive. When pure lines are crossed from the parental generation, in F1 generation you are getting pure lines of the parental generation. One is homozygous dominant and the other is homozygous recessive. You will get heterozygous dominant in F1 generation. Heterozygous dominant in F1 generation. When this F1 generation is self-crossed or self-pollinated, you will get 2 varieties in F2 generation. Okay, and one statement you have to remember here, beta, all the offsprings of F1 generation are phenotypically resembling the dominant parent, that you have to remember. And in F2 generation, after self-pollination of F1 generation, what you will get? This capital T, capital T, capital T small t and small t, small t. So if you have to consider the phenotypic ratio, you can get 3 dominant and 1 recessive, that is phenotypic ratio. If you have to take the genotypic ratio, 1:2:1, that is the genotypic ratio, where 1 is dominant, 2 are heterozygous dominant and 1 will be homozygous recessive. So, the phenotypic ratio is 3:1, genotypic ratio is 1:2:1 with respect to monohybrid cross. Now, from the monohybrid cross, Mendel has given us two laws beta. What are they? Law of dominance and law of segregation. Law of dominance says that factors occur in pairs. Mendel has called genes as factors. So, he says that factors occur in pairs wherein one is dominant and the other is recessive. The dominant allele is expressing and suppressing the effect of the recessive allele is what said from law of dominance. But law of dominance is not universal. It is not universal. Keep this statement in your mind. Why law of dominance is not universal? It is because law like incomplete dominance and codominance, they in that case in incomplete dominance, both the traits, both the alleles are expressing or the dominant allele is showing incomplete expression on the recessive allele. Now, we are going to see incomplete dominance as well. As incomplete dominance, codominance, pleiotropy, these have come into picture. That is why law of dominance is not universally accepted. When it comes to law of segregation, law of segregation which says that when dominant allele and recessive allele are migrating, both are independent of each other and two alleles are expressing, they express without any blending. They express without any blending. They express without any blending of alleles. That is the law of segregation. Without any blending, two alleles will segregate and they will express. Law of segregation is universal. Law of segregation is universally accepted. So, keep these two laws in your mind. So, now when we look into the test cross, before going to the exceptions of Mendelian, principles from monohybrid cross, first we will look into the test cross and back cross, which is very important. We can expect a question from this beta. So, yes, in the F1 generation, just now we have seen all the F1 offsprings are similar to the dominant parent, right? So, if you have to consider this flower color, if you have to consider this flower color over here, see, this is the F1 generation, F1 generation. So, this F1 generation all are resembling the dominant phenotype. They are resembling dominant parent, or you can say dominant parent. Now, we have to know the genotype of this F1 generation. So, test cross is nothing but the dominant phenotype crossing with the recessive parent. For example, here the dominant phenotype can be heterozygous or homozygous. When you are crossing it with the recessive parent, this will give you the genotype of the test organism. This will give you the genotype of the F1 generation. So, what is the purpose of test cross? To know the genotype and phenotype, mostly to know the genotype of the F1 generation. So, yes, when you cross like this, if you get 1:1 ratio of genotypic and phenotypic ratio, where 2 are white and 2 are purple. As such, if you get 1:1 ratio, then it says that the F1 generation is heterozygous. If you are getting all are heterozygous condition whereas all are purple, then it says that the F1 generation is homozygous dominant. Okay. So, this test cross will tell you whether F1 generation is heterozygous dominant or homozygous dominant. If it is heterozygous dominant, then you will get 1:1 ratio. Clear? Same thing, test cross can be conducted for monohybrid or dihybrid crosses. If it is heterozygous condition for monohybrid cross, you will get 1:1 ratio. For dihybrid, you will get 1:1:1:1. Dihybrid test cross ratio will be like this. Both the genotypic and phenotypic ratios are 1:1 only. Keep that in your mind. What is back cross? Back cross is nothing but F1 generation crossing with any of the parent is called as back cross. Next, Now if we look into incomplete dominance, first we have to list out what are the exceptions of Mendelian monohybrid cross, or what are the exceptions of Mendelian law of dominance. Incomplete dominance, codominance, multiple allelism, pleiotropy. These are the exceptions of Mendelian monohybrid cross. What you have to remember from this? You have to remember the examples and you have to remember the phenotypic and genotypic ratios for sure. So now if you look into incomplete dominance, dominant parent, that is red, and recessive parent, that is white. Okay, dominant parent and recessive parent. So homozygous red is crossed with white, but in F1 generation is not resembling either of the parents. Okay, F1 generation is not resembling either of the parents, and F1 generation all are pink. Heterozygous condition all are pink. Means here, according to law of dominance, the dominant allele should suppress the effect of recessive allele. Here the dominant allele is incompletely suppressing the expression of the recessive allele. So this is incompletely dominant, that is why we are saying it incomplete dominance. This is observed in snapdragon, anterhinum, which is where it is called as anterhinum, snapdragon, 4 o'clock plant. So here if you look into the both phenotypic and genotypic ratios of F2 generation, it will be 1 red, 2 pink and 1 white. Both phenotypic ratio and genotypic ratio are same, that is 1:2:1 for F2 generation. Keep that in your mind. Second is codominance beta. Second is what? Codominance. When it comes to codominance, for codominance and multiple allelism, keep this example in your mind, that is ABO blood grouping, that is ABO blood grouping. Before getting into the ABO blood grouping in detail, first you have to know IA blood group or IA antigen over the RBC, H antigen will be present if it is having the enzyme N-acetyl glucosamine transferase, N-acetyl glucosamine transferase enzyme. This enzyme will add on the sugar, that is N-acetyl glucosamine, which act as A antigen. Fine. Then when it comes to IB blood group, that is over the RBC, you have H antigen which is coding for the enzyme N-galactose transferase, which is coding for the sugar galactosamine. This codes for B blood group. This sugar codes for B antigen and B blood group. So here this two you have to keep in your mind. IA allele, that is A antigen, N-acetyl glucosamine transferase, which is adding on to the A antigen, that is N-acetyl glucosamine. IB, that is B antigen, which is coding for galactosamine. Keep that in your mind. So now if you see completely how many phenotypes and genotypes are possible for this, and here you can see 3 alleles. There is only one character, that is ABO blood grouping. Okay, ABO blood grouping is one trait. Okay, that is blood grouping is one trait. This is one trait, but this trait is controlled by 3 alleles, that is IA, IB or you can say IO alleles. These are the 3 alleles. What are the combinations of blood group that you can see beta? You can see IA, IAIO, IBIB, IBIO. You can see IAIB and you can see O blood group again. So as such you can get how many phenotypes? A blood group, B blood group, AB blood group and O blood group. So, total genotypes you can see there are 6 genotypes, 6 genotypes and 4 phenotypes. Keep that in your mind, ma, very important. 6 genotypes and 4 phenotypes in ABO blood grouping. So, this is kept under multiple allelism, as this one trait is controlled by 3 alleles, more than 2 alleles. According to Mendelian law of dominance, each factor has 2 alleles. But here you can see this one gene or one factor or one trait is having more than 2 alleles. That is why this example is kept under multiple allelism. Second is what, ma? It is kept under codominance. Why is it kept under codominance? Because if you see AB blood grouping, here A gene, that is A antigen is expressed, B antigen is also expressed. So, as this is kept, as the both alleles are expressed in the AB blood group, that is why this is kept under codominance. Both the dominant alleles are equally dominant to each other. That is why it is kept under codominance. So, with this incomplete dominance and codominance, multiple allelism is done. How is it deviating Mendelian law of dominance? Now, pleiotropy. What is pleiotropy? If one gene is showing more than one expression, okay, one gene is showing multiple expressions, you can say multiple expressions, or you can say multiple effects, that is pleiotropy. So, in pleiotropy, if you consider, ma, best examples include starch synthesis, phenylketonuria, okay, starch synthesis, phenylketonuria and skin pigmentation of Drosophila, white eye mutation of Drosophila. That is also one example for pleiotropy. How starch synthesis is related, this one we will discuss. It is related as such key. So, here if you see capital B, that is homozygous dominant condition, capital B small b, that is heterozygous dominant, capital B small b, that is again heterozygous dominant. Here you have to take small b, yes. So, if you see capital B, so large starch grains are present and this is round. Capital B small b, intermediate starch grains, starch synthesis, and these are round. So, capital B small b here is also showing incomplete dominance, So, if there is a question in the starch synthesis, which combination is showing the incomplete dominance, then you have to go for heterozygous dominant. Small b, it is again showing small starch grains and wrinkled. So, multiple expressions are shown by one trait or one gene, that is starch synthesis. How phenylketonuria is related? Phenylketonuria is Mendelian, autosomal Mendelian recessive disorder. So, yes, phenylketonuria is caused when the aromatic amino acid, that is phenylalanine, when it is not converted to tyrosine for its expression, then phenylketonuria disease is seen. This is not converted because of the mutation in one enzyme, that is phenylalanine hydroxylase, okay, phenylalanine hydroxylase, this is not expressing, that is why phenylketonuria is seen. Just by the mutation or not expression of one enzyme, how many effects are seen by phenylketonuria is multiple effects like frequent micturition, kidney poor absorption and mental retardation, skin pigmentation, all these effects are seen just by mutation of one enzyme, and this phenylalanine will be stored in the form of phenylpyruvic acid. This is stored in the form of phenylpyruvic acid. So, just absence of one enzyme or mutation in one enzyme is giving out many effects like poor absorption of kidneys, frequent micturition and mental retardation, skin depigmentation, all these effects are seen. And now if you see Mendelian dihybrid cross, where two traits are considered over here in Mendelian dihybrid cross. In our academy, the example which is taken is seed color and seed shape. Okay, seed color and seed shape, these are the two traits considered, that is why it is Mendelian dihybrid cross. Now if we have to quickly go for the examples where we can see or the numerical part, what we can expect from the dihybrid cross, you have to remember this 9:3:3:1 ratio. I will tell you one superb trick to solve any of the numerical question that is coming from dihybrid cross. Listen carefully beta. One amongst 9 these are round and yellow, these are wrinkled and yellow, 3 and 3 are round and green, and 1 is wrinkled and green. This we all know, right? So yes, 9 if you take round and yellow, what will be its combination? One is homozygous dominant, one is homozygous dominant, and one is homozygous recessive. One is homozygous dominant and the other is homozygous recessive. 4 are heterozygous dominant. 4 will be heterozygous dominant. And next 2 will be in this combination, 2 heterozygous and 1 homozygous, and here 2 are heterozygous, 1 is homozygous, like that. These are the combinations that you will get from 9 dominant, that is round and yellow. Out of this 3, what you will get? 1 will be homozygous condition and 2 will be homozygous recessive and heterozygous dominant. Same applies to this also. Okay, 1 will be homozygous condition and 2 will be, one is heterozygous and the other is homozygous. This is the combination of the genes that you get. Now, for example, in your question, amongst 560 seeds, how many are homozygous, 1 recessive and 1 dominant? As such combination if it is asked. Out of 16 zygotes, 2 you will see, out of 560 how much? Like that you have to calculate. For that, this should be in your mind. Okay. Now, when it comes to polygenic inheritance, what is polygenic inheritance? Many genes are controlling one trait. Okay, many genes are controlling one trait, that is polygenic inheritance. In polygenic inheritance is otherwise called as quantitative inheritance, because dominant alleles will show additive or cumulative effect. Here dominant alleles will show additive or cumulative effect in the polygenic inheritance. Best examples for polygenic inheritance that you have to remember is skin color in humans, height in humans, and kernel color in wheat. These are the examples for polygenic inheritance. So now if you can picturize the 3 laws and its exceptions, what are the 3 laws that are given by Mendel? Mendel's law of dominance, law of segregation. Of that, law of segregation is universal. From dihybrid cross, which law is proposed beta? Law of independent assortment. What is law of independent assortment? At the time of gamete formation, the movement of one allele is independent of the other allele. That is law of independent assortment. For all these 3 laws, we have deviations or exceptions for this. What are they? Incomplete dominance. What example we have looked into? Snapdragon or Antirrhinum. Codominance, ABO blood grouping. Multiplalism also, ABO blood grouping. Pleiotropy, phenylketonuria, starch synthesis, Y-tie mutation in Drosophila. Next, polygenic inheritance. Polygenic inheritance, best examples that we have looked into is kernel color in wheat, skin color and hair color in human, or height also in humans, like that. These are the examples for polygenic inheritance. And this chart you have to remember with respect to phenotypic and genotypic ratios. Very important beta. If you are unable to remember now, I will tell you a good trick for dihybrid and trihybrid cross. What you can do? Just square on 3:1. You will get dihybrid cross. Square on 1:2:1, you will get the genotypic ratio of dihybrid cross. Like that you can remember. And test cross we can know, that is 1:1. Incomplete dominance and codominance will be 1:2:1, both for phenotypic and genotypic ratios. This you have to keep it in your mind, ma. Ratios are very important. And now comes the calculation part, which is utmost important. Types of gametes. How you have to calculate the types of gametes? For example, there is a genotype that is given to you, this. Okay, and this is given to you, and they have asked you to identify how many types of gametes are possible from this genotype. Okay, how many types of gametes? For types of gametes, the formula is 2 to the power of n. That is the formula, right? So when 2 to the power of n is the formula, n will be number of heterozygotes. What is n? Number of heterozygotes. Here how many heterozygotes are there? 1 and 2. So 2 to the power of 2 will give you 4. Okay. How many types of gametes are possible from this? 4 types of gametes. That is the type of gametes. How you can identify number of zygotes or offsprings that are formed? Number of zygotes or offsprings, the formula is gamete square. Once you are identifying the types of gametes, just square on those number of gametes, you will get number of zygotes. Like we have found out the 16 zygotes from the dihybrid cross, or 4 zygotes from monohybrid cross in F2 generation, like that. And how you have to identify number of phenotypes and number of genotypes? This is the formula, 2 to the power of n and 3 to the power of n. For multiple allelism, to identify number of phenotypes and genotypes, this will be the formula. For polygenic inheritance, this is the formula. Genotypic ratio for polygenic inheritance and normal number of genotypes is same, that is 3 to the power of n. This formula chart, if you want, you can take a screenshot of it beta, and just to remember it, we can expect a numerical question from genetics for sure. Okay, so you can take a screenshot of this. Now comes the concept of chromosomal theory of inheritance. Who are the scientists that gave the chromosomal theory of inheritance? It is given by Sutton and it is given by Walter Sutton and Theodore Boveri. Okay, Sutton and Boveri gave the chromosomal theory of inheritance. They just united the chromosomal segregation with Mendelian's principles. What did they do, ma? They unite chromosomal segregation with Mendelian principles, and they gave us the chromosomal theory of inheritance, stating out 3 points which are important. One is chromosomes and genes, they occur in pairs, and segregation at a time of gamete formation is seen. Same segregation at the time of gamete formation, gene is also seen, because it is simple, right? Where are genes present? Genes are present in the chromosome. Segregation of chromosome will give you segregation of genes as well. Independent pairs segregate independently of each other. One pair will segregate, or one pair is independent of the other pair in genes, like that. Now, to prove, who gave the experimental proof for chromosomal theory of inheritance? That is Thomas Hunt Morgan, taking the model organism Drosophila. Why did he take Drosophila as the model organism? Because single mating can give you thousands of the flies, and to culture fruit fly it is very easy on the laboratory, and there are clear distinguishing features between male Drosophila and female Drosophila, where female Drosophila has ovipositor like this. Taking these criteria into consideration, Drosophila is chosen as the model organism. Not just chromosomal theory of inheritance, linkage, crossover, sex linkage and crisscross inheritance, all these are discovered by Thomas Hunt Morgan itself. Who is the scientist who gave crossover, linkage and crisscross inheritance? Is Thomas Hunt Morgan. That is why he is called as flyman of genetics. And now here comes the parental combinations and recombinant combinations. Before understanding this cross A and cross B, this picture should be in your mind. That is very easy. Consider this. Consider this as X chromosome from female, and this is the Y chromosome from male. Of course, this is shorter than X. That is why I have picturized like this. So now if you have to consider body color, eye color and wing shape. Body color, eye color and wing shape. Body color, dominant is what? Brown color, recessive is yellow. So here Morgan, how did he name it, ma? Mendel has named the alleles with respect to dominant allele. Morgan has named it with respect to recessive allele. For dominant, he has added on the plus sign, like that you remember. For eye color, which is dominant? Red eye is dominant, white eye is recessive. The name is given as white. Here for red, it is W plus. Same, miniature wing is recessive, normal wing is dominant. This is M plus and this is M. Okay. So, like this he has given. Now, if you see, the distance between these two genes is less, and body color and miniature ring, eye color and wing shape, these are far. One thing you remember, the distance between two genes on a chromosome will decide the recombination frequency. If the distance between two genes is more, then the recombination frequency is more. Recombination frequency is directly proportional to the distance between two genes. Keep that in your mind, okay? Recombination frequency is directly proportional to distance between genes. Distance between genes. Fine. So, recombination frequency is directly proportional to distance between two genes. Distance between body color and eye color is less. So, in cross A, when these two are considered, that is body color and eye color, that is yellow and white, and here dominant, that is brown body color and red eye color. When they are considered, of course, when you take the recombination frequency, how you will take the recombination frequency beta? Number of recombinants by total progeny into 100. That recombination type when you take, it is just 1.3%, and the parental combinations you can see 98.7% of parental combinations. It is because distance between these two genes is very less. Here if you see, distance between these two genes, that is eye color and wing shape, these two genes distance is more, which is saying that recombination types here are 37.2. Okay. One point here you have to remember, maximum recombination frequency or recombination type will be just 50%. Maximum recombination type is just 50%. So, here the recombination type is 37.2 because the distance between two genes is more, and parental type is 62.8. This you have to remember. If this is in your mind, understanding this diagram is just very easy. Now, chromosomal mapping. Chromosomal mapping is given by Alfred Student, wherein the formula is total number of recombinants by total number of progeny into 100. This is the first chromosome map he has given on Drosophila melanogaster. Here how you will identify the distance between two genes? Between two genes the distance is measured as centimorgon or 1 map unit, okay, or 1 map unit. So, if it is recombination percent is 5%, then the map units are how much distance you will cover? It is the map units are 5 map units. Recombination frequency is directly proportional to the gene distance, that is in map units. Now, comes the concept of sex determination, which is very important. I will tell you what you have to remember here. So, sex is determined, that is gender is sexual characteristics of an individual is determined based on 3 criteria. One is environmental factors, second is genetic mechanism, and third is haplodiploid mechanism. Best example for environmental factors is turtles. Best example for turtles. In turtles, temperature will decide the gender of the turtle. At the time of hatchling of egg, if the temperature is less than 27 degrees Celsius, they turn out to be male. If the temperature is more than 30 degrees Celsius, they turn out to be female. This is how temperature or environment is controlling the gender of turtles. Fine. Influencing the sexual characteristics of turtles. Second is genetic mechanism. In this we are going to learn the male heterogamety and female heterogamety. We are going to discuss the examples as well. Haplodiploid type, example is honeybees. Haplodiploid type of mechanism, wherein diploid individuals which has complete set of chromosomes, that is 32 number of chromosomes, we are calling it as females. only half the set of chromosomes, that is 16 number of chromosomes, will turn out to be males. Okay, this is haplodiploid type of sex determination. And next, here if you see the genetic mechanisms, what like male heterogamety, that is wherein male is deciding, the chromosomal combination of male is deciding the sex of the individual. If there is only one X chromosome, and first I have to tell you about the scientist Henking, who gave us the knowledge about X bodies. Okay, he said he has conducted experiments on the spermatogenesis of insects and said that only 50% of the sperms of the insects are having X chromosome. Only 50% of them are having X chromosomes. That is what he has said. So, XX, XO type and XY type, these are two combinations of male heterogamety. XO type, if you see beta, examples include bugs, cockroach, I will use another color, include bugs, cockroach and grasshopper. And XY type, we all know it. What are they? Humans and Drosophila. Fine. Next, here if you see, female combinations are giving you the female heterogamety like ZW and ZO. Okay, based on the combination of female chromosomes, it is giving you the female heterogamety. Females are deciding the sex, female chromosomes are deciding the sex of the individual here. ZW, this examples include for this type ZW, ZZ are birds, fishes and reptiles, birds, fishes and reptiles. And ZO type, if you see, bugs, okay, sorry, butterflies and okay, butterflies you consider as of now. Okay, so yes, these are the examples of male heterogamety and female heterogamety. All the examples are important, ma, keep that in your mind. Now comes the concept of Mendelian disorders. Everything I will discuss in just 5 minutes. Remember it, like autosomal dominant. Autosomal dominant disorders include myotonic dystrophy. Myotonic dystrophy. And this disease is expressed as it is autosomal dominant, which is following Mendelian pattern of inheritance only, right? That is why they are included under Mendelian disorders. It can be expressed in the heterozygous condition or homozygous dominant condition as well. When it comes to autosomal recessive disorder, this disease is expressed only in the homozygous recessive condition. For example, best examples for this are color blindness and hemophilia. Color blindness and hemophilia. Fine. Next, X-linked dominant and X-linked recessive disorders, if we have to take. X-linked recessive disorders include color blindness and hemophilia. Here, autosomal recessive disorders which are expressed in homozygous recessive conditions. The diseases list is huge: phenylketonuria, thalassemia, cystic fibrosis, fine. So, these are the diseases, and last but not the least, that is sickle cell anemia, which is also called as point mutation. And here recessive disorders, these are color blindness and hemophilia, which is expressed in the homozygous recessive condition only. In females though, the disease should be on both the chromosomes. In males, if it is on one chromosome, then disease can be seen. Y-linked diseases, the disease is on the Y chromosome and this is expressed in only males. So, this is all about Mendelian disorders that you have to know. Fine. And now when it comes to the chromosomal disorders, okay, what are the chromosomal disorders that you have to know, ma? 3 chromosomal disorders: Down syndrome, Klinefelter syndrome and Turner syndrome. Down syndrome is due to the trisomy of 21st chromosome. Klinefelter syndrome is due to the trisomy of an allosome. Klinefelter syndrome is observed in males. Turner syndrome is seen in females. This is due to the monosomy of the allosome. Both the individuals in whom Klinefelter syndrome is seen and Turner syndrome is seen, both of them are sterile. Okay? Keep that in your mind. Now, pedigree analysis. To analyze the pedigree chart properly, we can expect a pedigree question also, which is important. We have to know how to identify autosomal dominant, autosomal recessive disorders, and we should be in a position to identify sex linked disorders as well. So, for autosomal dominant disorders, first and foremost thing beta, whether it is autosomal dominant or allosomal dominant, if there is no skip of generation, then there is a possibility for dominant disorder. It is as simple as it is. Okay, there should not be any skip of generations. And here affected individuals can have at least one affected parent, okay, that you can consider. And affected parents can sometimes have in the F1 generation unaffected children. These are the possible identification traits where you can identify like autosomal dominant disease. Just by identifying, you can't go for choosing option. After looking on to these traits, just apply and see, apply the alleles and see, and then go for marking the option. Okay? Just this will give you 80% of confirmation in analyzing the pedigree chart. After that, you have to go for alleles checking. Here in the autosomal recessive disorder, how you will identify? Definitely there will be a skip of generation, and unaffected parents can have affected offsprings. Next, X-linked dominant, again there will not be any skip of generation. Disease is never transformed from a father to a son. Disease is never transferred from a father to a son. All daughters of the affected father will be affected, because if the father is affected, X chromosome is affected, and in heterozygous condition also, if this is the condition in the females, then it will be affected. That is why all the daughters of the affected father will be affected in X-linked dominant disease. X-linked recessive, mostly the disease is transferred from carrier mothers to the affected sons. Like that the disease will be transformed. Both in the X-linked dominant and X-linked recessive, you can clearly see crisscross pattern of inheritance, that is transfer of disease from grandmother to granddaughter through the son, from grandfather to grandson through a daughter. That is crisscross pattern of inheritance. So, yes, disease is never transferred from father to son here, and they show crisscross pattern of inheritance. Skip of generation can be seen, and here mostly from carrier daughter the disease is transferred to the son. Like that you can remember for X-linked recessive. So, with this we have finished the concept of genetics. In coming 3 to 4 sessions, I am planning you the most expected questions from the all chapters of your Telugu Academy 1 and 2, where you will get complete questionnaire session. So, don't skip or don't miss those sessions at all, which will be very helpful in your last minute preparation for AP EAPCET beta. So, yes, don't get tensed, be confident and happy with your preparation, be extremely confident, don't be doubtful even single %, you will definitely crack the exam, and trust these questionnaire sessions which you are going to see in the further sessions. Thank you.